Hello friends and good morning to all of you. I am Asim Karketta from Department of Biotechnology, Sant Gairaguru Vishwavidyalaya, Ambikapur, and I will be speaking on antigen processing and presentation. Right? This topic belongs to the course Immunology from the MSc Biotechnology second semester. So these are the learning objectives after the completion of this lecture. On completion of the lecture, you are expected to have a basic idea about antigens and its types, about the processing pathways of different antigens, including the non-proteinaceous antigens, and the outcomes of antigen processing and presentation. To begin with, let's have an overview of antigens and antigen presentation. Antigens are substances which are recognized by immunoglobulin receptors present on the surface of B cells. These immunoglobulin receptors on the surface of B cells are actually membrane-bound antibodies. Or the antigens may be substances which can be recognized by T cell receptors when they are presented along with MHC molecules. Here, I want to make clear that antigens and immunogens are technically different because antigens are the substances which can only combine with the end product of immune response, basically the antibodies or the T cell receptors. On the other hand, immunogens are the substances which have the tendency or the capacity to generate an immune response. This way, we can say that immunogen can act as antigen, however, the reverse of the same is not true. Epitopes are the immunologically active regions on the surface of an antigen that binds to antigen-specific membrane-bound receptors present on the surface of lymphocytes or to the secreted antibodies. These are also known as antigenic determinants. Now there are a range of different types of antigens. For example, we do have proteinaceous antigen. We do have antigens which are polysaccharide, which are lipids, and also we have nucleic acid based antigen. The antigenicity of these different types of antigen varies. By far, the most prominent or the majority of the antigens are proteins. Now, the way in which the B and the T cell interact with the antigen is quite different. If we talk about B cells can bind with free antigens in solution. On the other hand, if we talk of a T cell, then a T cell can only bind to or respond to the antigens which are presented to them by the MSC molecules. There are two types of MSC molecules, class 1 MSC molecules and class 2 MSC molecules. Class 1 MSC molecules, these are present on the surface of all nucleated cells. However, class 2 MSC molecules are present basically on the surface of antigen presenting cells. For example, dendritic cells, B cells, etc. Okay. So, altered cell cells such as cancerous cells, aging body cells, or allogenic cells from a graft can also act as antigen and elicit an immune response. Antigen processing and presentation refers to set of processes occurring within the cell and resulting in fragmentation of antigenic proteins, association of peptide fragments so generated with MHC molecules, and the display of peptide MHC conjugate at the surface of presenting cells to be recognized by the T cell receptors present on the surface of T cells. Antigens at this point are of two types: endogenous antigens and exogenous antigens. Endogenous antigens arise within the cells and are actually normal cellular proteins, tumor proteins in case of a tumor cell, or viral or bacterial protein in case of a virus or bacteria infected cells. These are processed within the cytoplasm and are complexed with class 1 MSC molecules and presented to TC cells. Exogenous antigens, on the other hand, are the antigens which are internalized within the cells by the process of phagocytosis or receptor-mediated endocytosis. These are processed by endocytic pathway, and the peptide fragments so generated after the processing is over is complexed with class 2 MSC molecules and finally presented to T helper cells. Cells which display peptide in association with class 1 MSC molecules to TC cells are called as target cells, because these cells are the targets for cytotoxic T lymphocytes. Cytotoxic T lymphocytes, after activation, search for these target cells and eradicate them, while those cells which display peptides associated with class 2 MSC molecules to T helper cells are called as antigen presenting cells. Professional antigen presenters include dendritic cells, macrophages, and B cells. By far, dendritic cells are the most potent antigen presenters of all cells. The cellular components involved in presentation of exogenous antigens, from proteasome subunits to the peptide loading complex, are collectively referred to as antigen processing machinery, APM. Antigen processing pathways. Now there are at least two major types of antigen processing pathways, and they are: first, the cytosolic pathway, which handles endogenous antigens, and the endocytic pathway, which handles the exogenous antigens. Both of these pathways are similar in sense that they have evolved and are designed to process antigens within the cells. However, they differ in terms of the machinery they utilize for processing of antigens and the way they complex the processed antigens to the MSC molecules. In cytosolic pathway, we see that the processing is mediated mainly by proteasomes, especially the immunoproteasomes, and the peptide fragments so generated are complexed with class 1 MSC molecules. In endocytic pathway, we'll see that the processing of antigens is mediated by endocytic vesicles, mainly the lysosomes, and the processed antigenic fragments are complexed with class 2 MSC molecules. Here, one thing I would like to mention that most of the peptides so processed, most of the proteins so processed are never going to be conjugated with MSC molecules, but they will be degraded entirely into amino acids, and these amino acids would be utilized for building other proteins within the cells. Only a few proteins are actually going to be complexed with MSC molecules and will be finally presented on the surface of cells for activation of the T cells. So this is the schematic representation of the processing pathways. You see the endogenous pathway. In endogenous pathway, the antigenic protein has been produced inside the cell itself. It is processed through proteasomes. The processed peptides move into the lumen of rough endoplasmic reticulum, where they are complexed with class 1 MSC molecules, and through trans-Golgi network, these vesicles containing the antigen MSC complex finally are transported to the membrane surface, cell surface. On the other hand, in exogenous pathway, you see that exogenous antigens are first internalized. They are processed within the endocytic vesicles, the endosomes or lysosomes, after which the vesicles containing the processed antigens fuse with the vesicles processing the MSC class 2 molecules. Then finally formation of antigen MSC complex and their display onto the surface. So this is the schematic representation of both the pathways. You see both the pathways are similar, operational within the cell. However, the operation machinery is different. In endogenous pathway, we find the involvement of the proteasomes, then special transport proteins, the TAP proteins found on the membrane of rough endoplasmic reticulum. Whereas in case of exogenous antigen, we see the involvement of internalization process, then endocytic vesicles, lysosomes, and then there is no need of transporting the processed antigen within the rough endoplasmic reticulum. Rather, the MSC and antigenic fragments are complexed, associated within the fused vesicle itself. Okay. Let us now discuss the processing of endogenous antigens, that is the cytosolic pathway, in detail. Now, the endogenous antigens are degraded and presented with class 1 MSC molecules. The process of processing utilizes the same mechanism by which the cells maintain the normal turnover of intracellular proteins. As we all know that the levels of various cellular proteins inside the cell is strictly regulated. That is to say that the proteins are synthesized and maintained when they are required and are degraded, or their expression is switched off when they are not required. The degradation of the cellular protein is mediated by proteasomes. The proteasomes are the protein complexes, which are utilized for degradation of cellular proteins when the cell is not in need of it. The similar process is utilized for the processing and presentation of antigenic proteins via cytosolic pathway. The entire cytosolic pathway can be divided into 4 separate but related events. Firstly, the proteolysis of the endogenous antigens within the proteasomes, the transport of the peptide fragments so generated to the lumen of rough endoplasmic reticulum, association of peptide fragments with class 1 MSC molecules, and finally the display of antigen MSC class 1 complex onto the surface of a presenting cell. Now let us discuss the processing of endogenous antigens. The processing of endogenous antigens takes place within multifunctional protease complexes, which are known as immunoproteasomes. These immunoproteasomes are the proteasomes which are formed after the induction by inflammatory response. The normal version of proteasomes, which the cells usually have, are called as constitutive proteasomes. These constitutive proteasomes are responsible for maintaining the normal turnover of cellular proteins. The level of protein within the cell is strictly regulated. That is to say that the proteins are expressed and maintained only when they are required. Otherwise, the expression is switched off and the cellular proteins are degraded by the proteasome complex. The degradation process depends upon ATP and the proteins to be degraded are first marked by small proteins which are known as ubiquitin. Induction of the cells by cytokines, mainly interferon gamma or tumor necrosis factor alpha, leads to the replacement of three catalytic subunits of 20S component of normal proteosomes by the homologous versions B1I, B2I and B5I, and replacement of the 219S regulatory subunits with PA28 alpha and PA28 beta, leading to the formation of immunoproteasomes. These immunoproteasomes are specialized for handling the antigenic peptides. They have distinct peptidase site and they preferentially cleave the antigenic proteins within hydrophobic regions and thus generate peptides that terminate in hydrophobic. Such peptides are suitable for binding within the pits of MSC class 1 molecule. Thereby, these proteasomes serve an important role in antigen presentation. Also, the size of peptide fragments generated after processing is appropriate for binding within the pit of class 1 MSC molecules. For proteolysis, the proteins, especially the antigenic proteins to be degraded, are first marked by ubiquitin. The ubiquitin antigenic protein complex is then directed towards the central tunnel of the proteasome complex, within which the proteins are degraded and the peptides are released. So here you can see the overall assembly of the protease complexes. Okay, so this one over here is the constitutive proteasomes. The constitutive proteasome is comprised of a centrally located 20S core component, which is bordered by 19S cap region, right? One cap at the top while the other at the bottom. Now, if you see the core component, then it is comprised of 4 rings, right? 2 rings made up of alpha subunits, right? Alpha rings, one at the top and the other at the bottom, while 2 rings made up of beta subunits, right? Beta rings at the center. Now, each ring is comprised of 7 subunits. Okay, alpha ring containing 1 to 7 subunit, similarly beta rings containing 7 subunits. Right? Alpha ring is responsible for interaction with 19S cap region. Okay, and the catalytic activity relies with the beta. Okay, and specific subunits which are involved in the proteolysis are beta 1, beta 2, and beta 5. Now, after induction by cytokines, especially interferon gamma and tumor necrosis factor alpha, the constitutive proteasome is converted to immunoproteasomes. Right? In immunoproteasome, you see, first of all, the cap region has been replaced by proteins PA28 alpha and PA28 beta, while there have been minor substitution in the beta ring as well. Right? You see, the catalytic subunits of the beta ring, especially beta 1, 2, and 5, have been replaced by beta 1I, beta 2I, and beta 5I in immunoproteasomes. Okay, so these subunits they are more, prominently handling antigenic proteins, breaking them into peptide fragments, the size of which is suitable for complexing with class 1 MSC molecule, as well as they generate peptides with hydrophobic ends, which are suitable for incorporating within the pit of class 1 MSC molecule. Okay. So this table just show the functions of several subunits. Firstly, you see the alpha subunit, they mainly interact with the regulatory cap region. Okay. Beta they are involved in proteolysis. Okay, so especially beta 1 in case of constitutive proteasome and beta 1I in case of immunoproteasomes, they have caspase like activity. Similarly, beta 2 and beta 2 1 they exhibit trypsin like activity. Beta 5 and beta 5 1 they have chymotrypsin like activity. Okay, then PA28 these have got ATPs activity and they catalyze protein unfolding, gate opening, and protein translocation into the central chamber of 20S core compound. Right? You see, the translocation of the proteins within the central channel is also dependent upon the ATP. Right? So here you can see the overall process of degradation or processing of antigenic protein. Right? First of all, the antigenic protein that has to be degraded must be labeled by small proteins known as ubiquitin. The process is carried out by ubiquinating enzyme and is dependent upon ATP. Right? Several units of ubiquitin are attached to the proteins which has to be degraded. Ubiquitinated proteins or ubiquitin labeled proteins are directed towards proteasomes, the central channel of the proteasomes, wherein they are degraded by the catalytic subunits of the beta ring of the proteasome complex. Now, next is this transportation of peptide fragments generated by immunoproteasomes to the lumen of rough endoplasmic reticulum. Now, why the peptide fragments so generated needs to be transported within the lumen of rough endoplasmic reticulum? The answer to this question relies in the fact that the antigenic peptide fragments are being generated within the cytosol, whereas the processing of MSC molecules, especially class 1 MSC molecules, is within the lumen of rough endoplasmic reticulum. So, to be complexed with class 1 MSC molecules, the peptide fragments need to be present within the same compartment, the lumen of rough endoplasmic reticulum, where MSC class 1 molecules are being processed. Strictly speaking, the antigenic peptide fragments has to be transported to the lumen of rough endoplasmic reticulum. The process of transport is mediated by TAP complex. TAP stands for transporter associated with antigen processing. It is a membrane spanning heterodimer consisting of two proteins, TAP1 and TAP2. These belong to a family of protein is known as ATP binding cassette proteins, right? And the versions of these proteins are well defined in bacterial cells and several other cells where they are responsible for the uptake of several solutes from the extracellular side towards the cytoplasmic side. Okay? So TAP1 and TAP2 protein, they each have a domain which is projecting inside the lumen of rough endoplasmic reticulum and an ATP binding domain which is projecting towards the cytosolic region, right, towards the cytosol. Now, TAP has highest affinity for peptides containing 8 to 10 residues of amino acids, and this size of peptide is also suitable for fitting in the pit of class 1 MSC molecules, right? Also, the TAP favors the transport of peptides which are having hydrophobic or basic, carboxyl terminal amino acid residues, right? So both of these fact favors TAP to transport peptides, especially derived from antigenic protein, towards the lumen side, and also it is favoring the transport of proteins which are appropriately going to bind with class 1 MSC molecules. The transportation by TAP complex is dependent on the ATP the energy released by the ATP hydrolysis. Okay? TAP1 and TAP2, these are polymorphic genes which map within class 2 MSC region adjacent to LMP2 and NLP7 within chromosome number 6. Now, TAP deficiency leads to several disease syndromes that are similar to both immunodeficiency and autoimmunity. Because if the TAP proteins are malfunctional or defective or non-functional, then the transport of processed antigen within the lumen of rough endoplasmic reticulum would be hindered, and henceforth their complexing with class 1 MSC molecules would be hindered, their presentation on the surface would be hindered, and because of this, the immune response mediated by T cells would be hindered. Also, development of tolerance, immunotolerance by the T cells may also be hindered, right? So, the overall, on an overall, we can say that deficiency of TAP may lead to disease syndromes which will be similar definitely to immunodeficiency or autoimmunity, right? Now, capsaicin binding increases steady state level of TAP, thereby allowing more peptide to be transported inside the rough endoplasmic reticulum's lumen. Okay. So, this slide shows the overall arrangement of TAP complexes within the membrane of rough endoplasmic reticulum. As can be seen, this TAP protein, TAP1 as well as TAP2, they are multipass transmembrane proteins, right? And they have got the domains towards the lumen of rough endoplasmic reticulum, that is the luminal domain, whereas the domain towards the cytosol, that is the cytosolic domain. The cytosolic domain also contains ATP binding region, wherein the ATP can bind, which can bind to ATP, right? Also, here it is depicted that a tapacin protein, which is a membrane-associated protein found within the luminal side of the membrane, if it associates with the TAP protein, then the stability of TAP protein is increased, resulting in more transport function of antigenic peptides and efficient antigen presentation. Let's now see the association of antigenic peptide fragments that have been brought inside the lumen of rough endoplasmic reticulum with class 1 MSC molecules. Okay. Now, for the assembly of class 1 molecule itself, the presence of a short peptide within the binding groove of class 1 MSC molecule is required. Okay, that is, we can say that the antigenic peptide itself, first of all, guides the proper assembly of the class 1 MSC. Also, the overall assembly is mediated by molecular chaperone proteins. Basically, three chaperone proteins play critical role in proper assembly of class 1 MSC molecules. These chaperone include calnexin, calreticulin, and tapacin. Tapacin is TAP-associated protein. It can associate or it can interact with the TAP transport protein present in the membrane of rough endoplasmic reticulum. A fourth protein, which is known as endoplasmic reticulum protein 57, has also been found to be involved in the process of assembly of class 1 MSC molecule, but its exact role has not been deciphered yet. Okay. First of all, calnexin protein, that is a resident membrane protein of endoplasmic reticulum, associates with alpha chain of class 1 MSC molecule and promotes its folding. Slight folding of class 1 alpha chain increases its interaction with beta 2 microglobulin, upon which the calnexin protein is liberated, and the alpha chain beta 2 microglobulin now associates together and binds with calreticulin and tapacin. Tapacin binding brings the overall complex in the close proximity with TAP transporter, right? Physical association of alpha chain beta 2 microglobulin heterodimer with TAP promoter promotes peptide capture by class 1 MHC molecule before the peptides being transported are exposed to the luminal environment of endoplasmic reticulum. The complex of TAP, tapacin, MSC class 1, ERP57 and calreticulin is called as peptide loading compound. Peptides that could not be bound by class 1 MSC molecules will be rapidly degraded and be liberated within the cytosol. As a consequence of peptide binding, class 1 molecules displays increased stability and can dissociate rough endoplasmic membrane and exit towards the surface of the cell through Golgi net. So this is how the class 1 MSC molecules are being assembled. You see, first of all, class 1 MSC alpha chain associates with chaperone calnexin. The association with calnexin allows class 1 alpha chain to be folded partially. The partial folded protein now can associate with beta 2 microglobulin. The association of alpha chain and beta 2 microglobulin liberates calnexin and allows the binding of calreticulin and tapsin. The overall molecular assembly can now accept peptides which are being transported through TAP transporter, right? The antigen capture is mediated by tapsin as it can associate with the TAP transporter, bringing the TAP transporter and the MSC1 molecule in close proximity of each other. Antigen binding liberates the chaperone proteins and sets MSC1 molecule free to exit rough endoplasmic reticulum towards the cell surface through the Golgi network. So this slide shows the entire process of processing of antigen and its loading onto MSC1 molecule. See, the endogenous protein is being processed through the immunoproteasomes. Also, it is important to note that few proteins are completely degraded, liberating the amino acids, and few other they are just partially degraded into peptide fragments. These fragments can be transported within the lumen of rough endoplasmic reticulum via TAP promoter. Now, the TAP promoter can interact with the MFC1 molecule which is being processed through tapacin. The association of tapacin with TAP promoter on one end and the MSC1 molecule on the other end leads to antigen capture by class 1 MSC molecules. Once the antigen is being captured by class 1 MSC molecule, its assembly and folding and assembly is considered to be over, and it may be liberated and directed towards the surface of the cell. For the presentation of the peptide, it is assembled. Now finally, the display of MHC antigen complexes onto the surface of the presenting cells. Right? Now, once the loading of peptides onto MSC class 1 molecule is over, the assembly of class 1 MSC molecule is fulfilled and it is liberated from the lumen of rough endoplasmic reticulum and it enters the trans-Golgi network. Right? Within the trans-Golgi network, the post-translational modifications occurs. The post-translational modification extensively changes the N-glycans of the class 1 MSC molecules, resulting in the full maturation of the overall complex, which are finally directed towards the membrane and ultimately displayed onto the surface of a presenting cell. So this is how the overall process is accomplished in cytosolic pathway. You see, the endogenous antigens are processed by proteasomes, especially immunoproteasomes will be involved. The peptide fragments so generated are transported within the lumen of rough endoplasmic reticulum via TAP transporter. Within the lumen, these antigenic fragments, they bind with class 1 MSC molecules, which are also being processed within the lumen of rough endoplasmic reticulum. Once the peptide binds in the groove of class 1 MSC molecule, the assembly and folding of MSC1 molecule is thought to be complete, after which these MSC molecules will be liberated from the membrane, and the antigen MSC complex is now set free to move through trans-Golgi network to the cell surface, right? Once on the cell surface, the antigen carried by class 1 MSC molecule will be presented to CD8 positive TC cells, leading to their activation. After activation, the TC cells will be converted to cytotoxic T lymphocytes and will be killing all the target cells. Okay? Now we shall discuss the processing of exogenous antigens, that is the endocytic pathway. The endocytic pathway is so named because in this pathway the exogenous antigens are processed within vesicles which are known as endosomes. So that's why the process is known as endocytic pathway. This pathway is similar to cytosolic pathway, but is considerably different in terms of the machinery it utilizes and the type of MSC molecule which are being used to couple the processed antigens. Also, processing and presentation of antigen is done by special cells which are known as antigen presenting cells. Okay? The entire process may be divided into 4 separate but related steps or events. First, the internalization of antigenic protein or antigen itself, then fragmentation of internalized antigen within lysosome, association of peptide fragments with class 2 MSC molecules, finally, the display of antigen MSC class 2 complex on the surface of antigen presenting cells. Internalization of antigens. The very first step in endocytic pathway is internalization of antigens. This is so because the processing of antigens has to be carried out within the cells, whereas the antigens are primarily present outside the cell. So, before they could be processed, the antigen have to be brought inside the cell, and that's why internalization of antigens is the very first step in endocytic pathway. The cells which are processing exogenous antigens are specific and are called as antigen presenting cells. Professional antigen presenting cells include dendritic cells, macrophages, and B cells. Out of these 3 cells, dendritic cells are the most prominent antigen presenter as compared to macrophage and B cell, because macrophage and B cells needs to be activated by phagocytosis or by a co-stimulatory B7 molecule before they could act as an antigen presenting cell. In general terms, antigen presenting cells are those cells which can express class 2 MSC molecules on their surface and thus can present processed peptide antigen to T helper cells. Internalization or uptake of antigens by antigen presenting cells is mediated by endocytosis. Endocytosis in general refers to all the processes by which materials move inside the cell and is essentially of three types: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis, also known as cellular eating, is the process by which phagocytes uses projections of their cell membrane known as pseudopodia to engulf a large particle, giving rise to an internal compartment called phagosome. Pinocytosis, also known as cellular drinking, is a non-specific process by which cells engulf solutes which are dissolved in liquid drops by pinching in and forming vesicle smaller than phago. In receptor-mediated endocytosis, the cells will selectively uptake extracellular molecules if they can bind to the receptor protein on the coated pit, the cytosolic surface of which is covered by coat protein. Once bound, the coated pit on which the bound receptor protein is located invaginates inside the cell, pinches off, and forms a cycle which later on fuses with lysosome. So in this slide, we have compared the different antigen presenting cells, especially the dendritic cells, macrophages, and B lymphocytes. You see, the dendritic cells, they are the most potent of all the antigen presenting cells because they can uptake the antigen either by endocytosis or by phagocytosis. The class 2 MSC expression on their cell surface is constitutive in nature. They also show B7 molecules which are required for co-stimulatory activity, and they can activate naive T cell, effector T cell as well as memory T cells. Rest of these two cells, they lack many of the properties which were shown by the dendritic cells. This slide shows several methods of endocytosis. The first one is phagocytosis, which is a very well-known and friendly process in which you see the membranes, they surround the material, the particle which has to be internalized. This particle could even be a virus particle or even a bacterial cell. To be internalized, the membrane projects outside, surrounds the particle which has to be internalized. These projections are called as pseudopodium. Pseudopodium grow all around the particle to be internalized and meet on the opposite end, thus internalizing the particle which is actually covered within the membrane. This internalized compartment is known as phagosome. Macropinocytosis, it's a process, actually non-specific process in which you see several solutes along with the liquid droplets are being internalized in the cell. Then rest of the three figures are depicting receptor-mediated endocytosis. In receptor-mediated endocytosis, we have several pits which are coated with receptors towards the extracellular side and towards the cytoplasmic side, towards the cytosolic side. These contain proteins. Okay? So these receptor-mediated endocytosis, this is a very specific process. Only those solutes which can bind with these receptors can be transported inside the cell. Fragmentation and internalization of antigens within the lysosomes. The antigens which have been internalized by the process of phagocytosis or receptor-mediated endocytosis is processed within increasingly acidic endosomal compartments. Right? The early endosomes, the pH of which may be ranging from 6 to 6.5, late endosomes, also known as endolysosome, the pH of which may be ranging from 5 to 6, and finally lysosomes, the pH of which may be ranging from 4.5 to 5. Now, acid-dependent hydrolytic enzymes degrade the antigens into peptides containing 13 to 18 amino acid residues. The peptides of this length or this size are the best fitting with class 2 MSC molecules. Internalized antigen move from early to late and finally to lysosome, encountering hydrolytic enzyme and increasing acidity within each compartment. The lysosomes contain unique collection of more than 40 different acid-dependent hydrolyase, proteases, nucleases, glycosidase, lipases, phospholipases, and phosphatases, which leads to the degradation and fragmentation of internalized antigen. Within the compartment of the endocytic pathway, antigen is degraded and the fragments of appropriate size binds with class 2 MSC molecules. Eventually, endocytic compartments or portion of them may return to the cell's periphery and the molecules or the receptors which they are carrying again reaches the membrane. Thus, the recycling of surface receptors or certain surface molecules can also be done via the endocytic pathway. So this slide shows processing and presentation via endocytic pathway involving B cells. Now you see the antigen binding receptors present on the surface of B cells are actually the membrane bound antibodies. The membrane bound antibodies bind to the antigen. Actually, these antibodies would be concentrated within the pits present on the surface of the cell. The cytosolic side of which is coated with clathrin protein. Once this, after binding with the antigen, this pit pinch in, forming clathrin coated vesicle, which fuses with early endosomes, where the processing of antigen is done. Part of the early endosome may again, part of the portion of the early endosome may again detach this larger endosomal compartment and be utilized for recycling of the surface receptors. However, the antigenic protein which was internalized now will be fusing with late endosomes and finally with the lysosomes, where they will be fragmented into peptide fragments of various size. Appropriate size peptide fragment may be complexed with class 2 MSC molecules and finally delivered towards the surface for display to T helper cells. Association of fragmented peptide with class 2 MSC molecules. Right? Now, unlike the cytosolic pathway, wherein the processed antigenic peptide binds with class 1 MSC molecule within the lumen of rough endoplasmic reticulum. In case of endocytic pathway, the binding of processed antigenic peptide and class 2 MSC molecule will be taking place within the vesicles outside the rough endoplasmic reticulum. So, until the MSC molecule reaches the vesicles within which the peptide fragments have been generated, the binding site of class 2 MSC molecules have to be masked away. Okay? So, to do this, within the rough endoplasmic reticulum, that class 2 alpha beta heterodimer binds with invariant chain within the peptide binding cleft of class 2 MSC molecule. Invariant chain plays an important role in preventing the binding of premature class 2 MSC molecule with endogenous antigens meant to be targeted to class 1 MSC molecules. Also, the invariant chain facilitates the folding of alpha beta chains of class 2 MSC molecules and the exit of the properly assembled complex from the endoplasmic reticulum, and it's targeting to endocytic compartments wherein they will be primed by the processed antigens. The invariant chain contains shorting signals in its cytoplasmic tail that directs the transport of class 2 MSC molecules from trans-Golgi network to the endocytic compartment. Proteolytic activity within the endocytic compartments degrades the invariant chain, leaving just a short fragment of invariant chain which is termed as CLIP, class 2 associated invariant chain protein. This CLIP protein is going to be engraved within the cleft of the class 2 MSC molecule until it is replaced by appropriate antigen that has to be present. So in lysosome, the HLA-DM, a vesicle membrane protein, replaces the CLIP within the class 2 MSC group with an appropriately processed antigens. Thus, the MSC class 2 antigen complex so formed move towards the membrane, gets stabilized, and finally present the antigen to the T helper cell. So this is how the class 2 MSC molecules are assembled and loaded with antigenic peptides. You see, within the lumen of rough endoplasmic reticulum, the alpha-beta heterodimer of class 2 MSC molecule, they bind with invariant chain protein, right? The binding of invariant chain with class 2 MSC molecule leads to the blocking of the groove of class 2 MSC molecule. This prevents the binding of endogenous antigen within the groove of class 2 MSC molecules. On the cytosolic side, this invariant chain also contains shorting signal, which guides the class 2 MSC complex to appropriate endocytic vesicles within which the antigenic proteins are being processed. Once the MSC class 2 molecules reaches the endocytic vesicles, the acidic environment and the proteins present, the degradative proteins present inside the endocytic vesicle degrades the invariant chain, leaving just the CLIP segment which still binds within the groove of class 2 MSC molecules. HLA-DM, it mediates the replacement of the CLIP protein, allowing the binding of peptide, processed peptide or antigenic peptide within the groove. Once bound with the antigenic peptides, the class 2 MSC molecules attains further stability and they are released and finally reach onto the surface for the display of the peptides they carry to their helper cells. So this slide shows the overall process of the endocytic pathway. You see, the exogenous antigens are being internalized within the antigen presenting cells in the form of phagosomes. The phagosomes, they fuse with endosomes and finally with lysosomes. The internal environment of these compartments with which the vesicle containing antigenic protein is being fused is very different. The acidity of the compartment is different and the overall enzymes, degradative enzyme present is also different. The combined action of acidic environment and the degradative enzyme leads to the fragmentation of the internalized antigens. On the other hand, the class 2 MSC molecules are being processed within the lumen of the rough endoplasmic reticulum. You see, to prevent the binding of the class 2 MSC molecules with any antigen other than the internalized antigen, the antigen binding cleft have been masked away by the invariant chain protein right? The invariant chain protein also contains sorting signal which guides class 2 MSC containing vesicle towards the endocytic vesicles in which the internalized antigen is being processed. Once the MSC class 2 containing vesicle fuses with the endocytic vesicle, the invariant chain is degraded and only the clip region is left behind. With the help of HLA-DM, the clip region is replaced with the processed antigen, after which the class 2 MSC molecules becomes stable and now ready to present now ready to move to the surface to present the antigen to CD4 positive T helper cells. Cross presentation of extracellular antigens. This is not a major pathway of antigen presentation. However, this has been evidenced and reported by several researchers. In cross presentation, the exogenous or extracellular antigens that have to be processed and presented by class 2 MSC molecules are processed and presented with class 1 MSC molecules to CD8 positive T cells. That is, we can say that cross presentation is the presentation of extracellular antigens along with class 1 MSC molecules. There are two possible pathways for cross presentation. The vacuolar pathway, in which the antigens are internalized within the endocytic vesicles. They are fragmented within the endocytic vesicles and they fuse with the vesicles containing empty class 1 MSC molecules derived from rough endoplasmic reticulum. Thus, fusion of the vesicles containing the fragments of exogenous antigen and the vesicles coming from the rough endoplasmic reticulum carrying the empty MSC1 molecules leads to the formation of MSC1 antigen complex, which are finally displayed onto the surface of the presentation. Cytosolic diversion pathway. In this, the exogenous antigens are internalized within the endocytic vesicles. They are processed and fragmented. The peptide fragments so generated, they leave the endosome or they enter into the cytoplasm, wherein they are processed with proteosomes and are transported to rough endoplasmic reticulum and get associated with class 1 MSC molecules being assembled within the lumen of rough endoplasmic reticulum, and finally they are directed towards trans-Golgi network to reach to the cell membrane, right? So this way the antigens which were destined to be presented by class 2 MSC molecules are now being presented by class 1 MSC molecule in a phenomena which is known as cross presentation. This cross presentation is necessary for inducing immunity against most of the tumors and viruses which actually occur in the peripheral tissues rather than occurring in the antigen presenting cells. So this is the pathway for cross presentation. You see, the exogenous antigen was internalized and they may follow the vacuolar pathway, or it may follow this cytosolic pathway. Okay? Processing and presentation of non-protein antigens. We know that fairly good number of antigens are non-proteinaceous in nature, right? But then they have also the tendency to be processed and presented to certain T cells, right? So these non-proteinaceous antigens are presented by members of CD1 family of non-classical class 1 MSC molecules to T cells which bear gamma delta T cell receptors. These gamma delta T cell receptors, they have the tendency to bind with glycolipid antigens. And The CD1 molecules, they are encoded by 5 genes located on chromosome number 1. Okay? The CD1 molecule can present lipid components of Mycobacterium tuberculosis, Mycobacterium leprae, and thus they can induce T-cell mediated response. The processing pathway is quite distinct, and they can occur in the deficiency of TAP transporter. Finally, what is the outcome of antigen processing and presentation? Right? The ultimate outcome of antigen processing and presentation is activation of subsets of T helper and TC cells, right? As well as other cells which have important role in mediating immune response, right? Activation of T helper cells leads to secretion of cytokines, which in turn leads to activation of several other cells to enhance the immune response. These several cells, which are activated by the cytokines released by T helper cells, includes the TC cells, the B cells, the macrophages, etc. The multitudes of these cells, after getting activated, they fortify the immune response and thus help the body to clear away any pathogenic invasion or their ill effect. The cytotoxic T lymphocytes, after getting activated, they wander around in the body and they kill all the cells which are displaying foreign protein or altered cells. Antigen processing and presentation also plays important role in inducing self-tolerance. For inducing self-tolerance, the T cells as well as the B cells have to be introduced with the body's self-antigen. Introduction with self-antigen is mediated via presentation of antigens along with the MSC molecules on the surface of the cells. Okay, so this presentation is also mediated as a result of antigen processing and presentation. Activation of macrophages, B cells, and multitude of other cells that take part in immune response is also the outcome of antigen processing and presentation. Antigen presentation, especially the cross presentation, plays important role in inducing immunity against many viruses as well as cancers. Effective presentation may lead to syndromes which are similar to immunodeficiency or autoimmunity. So that's all for today. Thank you very much.